Hello, everyone. My name is Ashoka Rajendra, and I'm a product manager at Benchling. I oversee our registry and inventory products. And today I want to talk to you about foundations for data-driven drug development. Benchling is a life sciences research and development software platform. We work with companies of all sizes to improve their scientist productivity through modern software. We work with 12 of the 25 top biopharmaceutical companies, emerging biotechs, as well as companies in a variety of industries, such as synthetic biology and agriculture. No matter the research type, we've seen the same core challenges. These companies are leveraging bleeding edge discoveries in biologics or chemistry, but they're also trying to leverage bleeding edge data science techniques like machine learning. A lot of you have been to talks like this in the past, where you want to understand how you can use machine learning and data science to improve your speed of discovery, but also lead optimization, process development, preclinical, all the way up to IND filings. All of you have been to talks like this and you're like, how do I actually get to this world? And why is this so complicated? I'll start by level setting. People's expectations of machine learning are sky high. You think you'll be writing lots of complex algorithms and running codes and generating new insights over the data sets that you've generated internally. The reality is that most of your time is going to be spent acquiring data and cleaning that data, and a very small portion of your time is going to be spent running code and writing code and devising complex algorithms. To put an example of this, in 2018, Novartis declared it was going to be a data science company. And about a year into those efforts, the CEO of Novartis noted that they grossly overestimated how much clean data sets existed and how difficult it was to clean these data sets and link all the data together. It's taken them years to just get to a point where they have good enough data to actually run the algorithms in the first place. So why is it so difficult to generate clean data sets? Biopharmaceutical research and development data is very, very complex. To illustrate my point, at the center, I've put a traditional candidate or molecule that you might be generating, an antibody. But I've also put in this word cloud all the different concepts and items that you would need to relate to this antibody. So it's not just about the molecule itself. The process by which you create this molecule is extremely important to track. What plasmids did you use? What cell lines were used to produce this? What were the growth conditions? What was the yield? But then there's intrinsic properties about the antibody itself. What is the DNA sequence encoded that encodes synthesize it? What are the CDRs? What are the variable regions? So on and so forth. So you not only have to get the intrinsic properties for each of these items, you also have to track the relationships between these items. So what's the solution? We have this complicated work, and in order to get to this world of data science, many of you have thought we need to embark on this digitalization journey. We need to put everything that scientists are doing into software. So you might say, okay, let's get a electronic lab notebook so people are typing things in instead of writing them out. Let's use desktop design tools so people are doing sequence design. For registering molecules and compounds and cells and viruses, maybe you maintain an internal database of all the different things that you're creating in the lab. And then for your physical inventory of all the stuff in your lab, that might be maintained on a shared spreadsheet or document that tracks where things are in freezers and containers and plates and so on. So one of the challenges about just putting everything into software is that you have highly educated users who are not software specialists. So they may be very, very well trained in one domain, but it's not their job to adopt a bunch of different tools and know how to tie them all together. In addition, you have complex and ever-evolving workflows. So you can't just set up the software and tools for them to use because the science will lead them in a different direction and things need to change. You also have the challenge of having a very, very deep domain. There are very few purpose-built tools for this kind of work. But what I wanted to do is kind of take you through a case example of how this digitalization has gone in another industry. We have a cautionary tale from electronic medical records where they had a similar problem. They wanted to digitalize everything that clinicians are doing in order to yield benefits such as applying data science for safety, consistency, and generating insights about the treatment of disease to improve clinical outcomes. But the process by which they went about doing this turned clinicians into the most highly educated manual data entry workforce in the world. The federal government has issued edicts to reduce the burden on clinicians of actually using the electronic medical record. As we go through the process of digitalizing the work that scientists are doing, we cannot repeat this same mistake with our scientists. So what's the right way to approach this problem? At Benchling, instead of starting on the bottom layer, which is the platform that produces all of the data that you can then analyze, we start at the very top layer. Benchling supports a wide range of research and development platform or research and development processes across many applications that sit on the same platform. So for us, we think high quality applications drive user adoption, which drive high quality data input. Each application, whether it's a notebook or registry, molecular biology tools, process management, or inventory, all sit on the same platform. So in the middle layer, all the data is unified. And we have a team that works with you to understand your use cases and map and configure the data models and processes to your unique scientific use case. All of that then allows the underlying platform where you might be generating data sets to get data in and out. But it starts with the foundation of high quality user applications and configurations mapping to your scientific workflows sitting on a unified platform to actually have the data aggregated together to do analysis after experimentation. So let's walk through a case study of doing this. We have a top 25 biopharma company doing antibody engineering. And when they came to us, they had multiple databases for different objects they're working with. So cell lines, plasmids, antibodies, and their antibody database had some plasmids in it. Their plasmid database had some antibodies in it, but that was all stored in a relational database. You also had freezers and inventory tracked in a mix of internal tools and spreadsheets and experimental results, some of which were uploaded to their lab notebook and some of which were recorded in Excel. So what you'll note is they actually did have a lot of software, but the problem is they had very, very poor adoption and this system was costing them millions of dollars to maintain. The first thing you need is the ability to accommodate the complexity of the data that scientists are producing. This data structure needs to be native to the kinds of things that your scientists are working with: cell lines, viruses, plasmids, DNA, RNA, and so on. It needs to also track attributes of these things, not just the raw sequence information, but all the attributes and constituent pieces. And importantly, this data model needs to be flexible. The science will change over time. The use cases will change over time. So it's not something you just set and forget and never change. You also need to be adaptive to changes in workflow. So you might have a purification protocol that has an initial iteration, which draws a certain yield. You then move to a second and third iteration where you're adding different steps in your purification protocol. You should expect that as a root assumption. And when we're working with this company, these changes need to happen on the order of days or weeks, not months and years. And a big challenge with a lot of software that people use to digitalize the work scientists are doing in the lab is that it can take an extraordinary amount of time to adapt to what scientists are actually doing if that changes. And once that's out of date, you will lose adoption of your actual tool. Lastly, I want to talk about unified data. So many companies will have databases for a variety of the data that they produce. They might have molecular biology data, genome sequencing data, in vitro studies, in vivo studies, external collaborators, and so on. So all of this ends up getting stored into a database, and oftentimes the strategy is that you can pool all of this together into some data lake. All you're really doing is throwing the problem over the wall and punting it to be solved later. Because what happens is that once this data is pulled together in some data lake, it's really more like a data swamp. The people who should be spending their time analyzing this data now have to go track down who made this data, what was the purpose behind certain ways in which they structure the information that they captured, and they're the furthest away from the actual recording of the information, but then have to make sense of the data that they're trying to analyze. A high quality application allows you to link data at the point of data entry. So this is a really key point. By having a super scientist friendly user interface, and what you see on the right here looks a lot like what Excel tables might look like, except at the moment that the scientist enters data, it is validated and tied together and linked together instantly so that you don't have to come back to it later and figure out what people meant or what people were trying to do. So by having this data entry format and all of this information being tied together at the outset, that then becomes queryable by an API or using SQL. So what we're showing here is something that'll be familiar to software engineers. So our underlying platform allows for your data scientists and software engineers to access the information inside of the system using the API. So every kind of item that you load into the system or have scientists create, the results that they record can either be programmatically put into the system or programmatically taken out of the system for downstream analysis. So let's kind of tie this all together. Without any fancy data science, machine learning, we were able to shut down 4 redundant databases, save the customer $4 million, dollars, and get a 90% increase in compliance and adoption. So this is only focusing on what are the applications that can make scientists more productive, consolidate their data into a single and unified platform, and allow them to do their work. So if you start with the premise of better data comes from having high quality applications, you will get data that is structured, unified, and portable that allows data to be shaped in a consistent way, tied all together, and then synced out into systems that can be used for analysis. So after doing all of this work, you can then use sophisticated analysis tools to actually run these fancy data science and machine learning algorithms across your data sets. So for scientists, this might mean understanding whether a candidate will fail in an in vivo trial by mining the in vitro assays. Managers might be able to look at, which candidate will advance to the next stage or might need more tests. And executives could look at this information and analyze it and see, what is the state of your early stage pipeline and are there risks that they need to weigh in on? I'd like to thank all of you for joining. You can visit us on our virtual booth and please feel free to ask questions or reach out. Thank you.